CAR T actually is a pull of the different of the T cell with the same CAR, these molecules. And they have a subtype we call CD8, CD4, and also under the CD4 they have different pull of the different kind of the T cell with different of the age, different of status. So in this case, different from the traditional drug, no matter small molecular and large moleculars, CAR T is alive. This is a alive drug, alive medicine. It means the cell themselves is they are patients by patients is they're different. They're different of the composition, different of the status. So in this case, if we use a different of the method to make the CAR T cell, then they will go to the totally different of the products. In this case, using the instruments, maybe will give a little bit better to make the cells with the more consistent of the process for the manufacturing. So in this case, we can use the hands to make it the CAR T cell. We can use different of the instruments to making the CAR T cell. However, because cell culture not only just make them alive, but also you have to make these cell expanding to more than 100 to 200 times to get to the high quality of the cell. CAR T, we know, is they composed by two major parts. One is a CAR, so company can design and to develop the new CAR. This is look like a weapon. T cell is usually is came from the patients in the allogenic in the same patients of the T cell. So in this case, these T cells look like the soldiers. They carry the one new weapon. This weapon name we call CAR. And in this case, we're making the whole of the CAR T, this new medicine, their performance not only just based on the CAR, also they're based on the T cell themselves, how these soldiers fighting with cancer cells. So we try to explore so many different of the instruments to try to get the more of the mimic and close to the cell culture with more of the controllable. In this case, if we use a bag or use a tank to culture the cell, and the cell, because of this volume, so they always have on the surface or in the middle of the distance. These cells in the different area of the culture system, they will show the different of the gain of the neutron and also gas. So in this case, you cannot control each cells. They will get same of the conditions and the neutrons. So in this case, photofab is one of the ideal of the we call the matrix to culture this cell, because your gas and the neutron just on the narrow fiber on the surface to go through. So in this case, the distance of each cell to get the neutron and the exchange of the gas, they are quite similar. So in this case, we can control the condition to make the CAR T cell is more of the controllable and more of the healthy. So this is why we use quantum this instrument, because the key, the beauty and the key of the mechanism is they can make the every cell same of the condition during the culture. And we can see, and this is one of the issues. So in this case, because they got quite a similar of the condition, we can control the cell growth. So in this case, we can shorten the cell culture to make the same sufficient of the CAR T cells to deliver to the clinical studies. This is one of the advantages. Secondly is because CAR T usually is early stage, they usually use hands to study and to develop the CAR T technologies to culture the cells. So in this case, the menu in the flask or in the dishes of the culture system can easily to move to the use the quantum and without any gap. So this is very important. You can see in my presentation in the conference, the data from no matter is a menu of the using the flask or using the dish, there are cells of the final of the CAR T cells. They are no matter they are CD4, CD8 of the ratio, and also their status quite similar. So in this case, the researchers don't worry about in the future of the clinical study. They can more easy to scale up the CAR T cell production, and then they can use in the clinical studies. The main, benefits or the advantages. One is this is a closed system. It means they will avoid contamination in the air. In the culture environment. So this is the first important. The secondly is you can scale up very easy, and you can based on the you can add the more of the column. You can scale up the cells within the same machine. This is also very convenient. So we call robust. And also, you can shorten the cell growth. Most of the leukemia patients, their conditions is very aggressive. In this case, waiting one more day or 2 more days, they will cause the patients without, they cannot waiting for you are finished the CAR T production, and then they already pass away. So this is why timing in this issue is very important to shorter it better. And so this is why I think this is the major issues. The fourth one, I already talked from the research scale to the clinical manufacture scale. This is almost no gap. So you can easily to scale up your production of the CAR Ts. So I think from the current situation, this is quite ideal of the culture system for the CAR T therapy. And so this is why I talked to the company also several times. Currently only the automation, but how to make all of the automation be more AI elegant, artificial elegant. This is the final of this machine have to go because we use the different of the patient's own cell, and their initial of the status and culture, the condition should be different. Some patients, maybe they feel very thirsty, so you should feeding of the water. Some patients cell, they will feel very hungry, then they will feel the bread. So this is need more of the feedback and using the AI systems. This is, I think, one of the direction of the instruments for the culture the cells. This is one of the necessary way to go ahead. Not only just closed automation and easy to handle and friendly of the software to allow you to handle these instruments, but also just I talked to you before, we need more of the smart instruments to based on the patient's cell or based on the cell culture conditions, they will automatically to adjust their feeding of the nutrient, medium, and some of the factors. This is, I think, the very important. Otherwise, you still have to stay beside instruments to monitor the cells growing, and then you have to adjust your culture conditions to feeding some of the different of the nutrients and cell mediums.